Thank you. Thanks, Phil. So I know I'm standing between you and lunch, so I'll try to be brief. And I'm Leandro. I'm the technical lead for the Cellular Analysis facility. And we are, why it's not, there you go. Now it's switching the slides. Sorry about that. So the Cellular Analysis facility is a facility that we support and partner with different researchers across the college, the MVLS. We have partnership and support for literally all the schools within college, but not only within the college, the MVLS, but also we support research other areas of the university, for example, the College of Science and Engineering. Here you're observing piece of a rock from the Isle of Iona. And you can see those lines, they act like three lines. So you can use those lines to trace the age of a particular rock. We also support projects from College of Arts and Humanities. Different students there, when their final project, they use our facility. For example, here you've seen an image from a vinyl record. And here on the right, an image of clay. So we use the SEM to in different kind of clay and see if there was a difference between them. But we are not only bound by the walls of the University of Glasgow, we also support different projects from other research institutes, other universities across Scotland and England, as well as industry partners. But what is the Cellular Analysis facility? So I lead a team of 8 highly skilled technologists, Tony and Susan around light microscopy, Margaret in the electron microscopy, Diane, Alana, and Daniel in the flow facility, Hannah in light microscopy intravital, and Tyler in histology. So the Cellular Analysis facility is formed by 4 cores. As you can see here in this drawing, we have the light microscopy, electron microscopy, flow cytometry, histology, and connecting these 4 cores, we have the bioimage analysis. So what I'm going to do today is briefly show some of the technologies, the techniques that we can support you on these different cores, and perhaps you can have an idea of all the different capabilities that we can provide to you. So in histology, we can do wax processing and sectioning, as well as cryo-sectioning of different kind of tissues, immunohistochemistry, new antibody optimization, immunohistochemistry training. We have a slide scanner to image your slides that were prepared in the facility. And here just some examples of the kind of how skillful is Tyler. We have here some special staining like Austrian blue of column. Double immunohistochemistry here is a piece of a bone marrow. And here's quite interesting, is a whole leg of a mouse that you can actually see here, the ankles and the bones. So it's really amazing. In terms of flow, we can provide support in flow cytometry and cell sorting. In flow part, you can use flow for example, detection of fluorescent reported genes, immunophenotyping, analyze cell cycle analysis, calcium flux, mitochondrial membrane potential, for example, and measure of cell proliferation. And in terms of cell sorting, we can support you up to 4 populations that can be sorted simultaneously, where a high concentration of target cells can be collected with over a 98% purity of cells. And from here, you can bring your sample, for example, to other facilities from for example, for John's or for Katie's, where you can do RNA-seq, metabolomics, lipidomics, or even get those cells and go cloning a particular clone that you might want to, or go to cell culture. So here's an example of the use of our sorting capabilities. A particular group that was just published at the end of the last year. They were interested in a particular subtype of DC dendritic cells from patients with rheumatoid arthritis, and they use different gatings up to getting those 2 different kind of DC subsets they were aiming for. And later they use a different gating to get memory T cells that they could culture together with those DC subsets, and later on they brought those kind of cultures then for single cell RNA-seq. So this is just to give an idea that our facility is not like within the shared research facilities. We are not like silos. We have bridges, connections between one another, so we can use part of our facility to then bring to another one. So these are some of the machines that we have in the facility. So in terms of flow, we have a conventional analyzer like the Canto, the Celesta, and the Fortesa. We in the last few years we have been successful in acquiring some investment from the university directly on the acquisition of some spectral analyzers. So different from the conventional ones, where in the conventional you use physical filters to select which fluorescence you want to capture. With spectral analyzer, you capture the whole thing and then use spectral unmixing to isolate the fluorescence, so that actually allows us to have a much more complicated panels, much complex ones. So it allows you to bring much more in-depth analysis of your samples. And in terms of sorting, we have two areas, BDF areas on it, and as well our new Sony MA900. As you can see here in this picture of the Sony, is within a cabinet that we can use to let's say more challenging and infectious samples that can be used for sorting. In terms of electron microscopy, we can use transmission electron microscopy, for example, to image nanoparticles, isolated proteins, for example, proteins that were that you want to characterize better in terms of the structure. You can use negative stain to see the structure before going, for example, for a more high-end machine like the cryo-EMs at the Glasgow campus. You can use the EM also to observe cellular structures. For example, here you can observe endoplasmic reticulum as well as mitochondria, and we can combine electron microscopy with immunolabeling. For example, here you're seeing an exosome that was labeled with some antibody here with gold, connected to gold. In terms of scanning electron microscopy, you can use that to see the topology of your sample. For example, it can be the morphology of a 3D matrix, as you can see here on the left, or see the difference on the morphology of infected and infected cell. For example, here, difference between erythrocytes that were uninfected or infected with malaria parasite, observe organized, in this case here in the middle, or even see the interaction between different organisms. For example, here, helminth that was infected the gut of a mouse, and here, Cryptosporidium oocyst in the intestine of a cow. 2 years ago, I successfully led a grant application to the MRC, where we update our electron microscope, our transmission electron microscope, and now we can also offer a volume EM. So here you can see the observation of a 3D observation of the nucleus and the closer proximity of the mitochondria with the nuclear pore on the cell, something that was kind of hard to observe in a 2D thin section of that is normal for transmission EM, but now with the volume EM, you can see much more clearly this interaction between the nuclear pore and the membrane of the mitochondria. 2 years ago, with the establishment of the shared research facility, we got some investment from direct investment from the college in the refurbishment of a basement area in one of the buildings in the main campus. This allowed us to build a bespoke imaging suite where we concentrate most of our high-end machines, making it easy access for the users to use those machines, as well as where we have the office of the facility staff here, making it easy this connection and this interaction between the users and the facility staff. So as you can see, it's quite a nice bespoke image suite that houses all these high-end machines. And together in this refurbishment, we also secured some money to buy some new instruments, for example, a high throughput system, a new live cell confocal, a new super resolution microscope, and a new multiphoto for intravital imaging. In terms of like microscopy, we can support live cell imaging that can be for a few minutes, for example, here in this mitochondrial dynamics, or can be even imaged for throughout different days. As you can see here, the development of a gametocyte, of a malaria gametocyte within the erythrocyte. In this imaging, we have like this machines, they have a heated stage and CO2 chamber, so they can allow live observation of your samples throughout different hours and days. We have different confocals that can be used to image a piece of tissues. For example, here, lung, synovial tissue, and the skin. Or it can also be used to image, for example, culture cells. We also have quite a unique machine that allows a tissue laser dissection. Again, like I said, we have different machines that can make this connection, this bridge between our facility and the other one. And this one, people are being used to remove a particular piece of the tissue and put in an epidoff, and later going to the KTs or John for further analysis, like metabolomics, lipidomics, or single cell, whatever. And this laser tissue dissection can be used, for example, piece of tissue that are being processed in histology, or it can be even a piece of tissue or even cells that you have in culture, and then you can collect that in epidoff and go for molecular analysis afterwards. This is quite a nice image, I think, because it shows the power of the imaging and how that can change our understanding of a particular experiment. So these are DNA fibers, and they were imaged here on the left with our standard confocal with these two different pools, magenta and green. And with using the confocal, you kind of think that the magenta and the green signal are overlapping each other. But when you use our super resolution microscope, our brand new one, you can actually see that the magenta and the green signal are actually parallel, not on top of each other. So that kind of gives a different interpretation of your results. So that kind of highlights how imaging and observing your sample can be quite important. In terms of intravital and in vivo imaging, we have different machines that are within the biological units that you can image the same mice, the same organism for different periods of days or even months. So here, I'll go back and back again because kind of. So here you can see the blood flow in the mice infected with gametocytes. They're expressed in green. Here in the middle, you have the observation of immune cells in a piece of tissue, and I think it was in the ear. And here you can see the development of a inflammation process within the same mouse over different periods of days. We have some high throughput technologies. We have the image stream, which is quite a unique machine. It's a combination between a flow cytometer and a microscope. So for each observed event that we are observing here, we can have up to 5 different images of it with different channels. So it combines that quite a unique observation of quantitative and qualitative analysis of your sample. And we also have the CX5 for high throughput, like 94, 384 well plays that can use for high throughput, and just connect to a workstation that can do the analysis of those images as you are acquiring them. And we can also combine different imaging technologies, for example, where we say correlative microscopies, where you can image a particular sample with light microscopy, where you can use these different multiplex capabilities of light microscopy, and later process the same area and the same sample for light microscopy. For example, here was scanning light microscopy, where we want to observe the same event that we can see here, highlighted in green, where in this case, we will want to see neutrophils, extracellular traps, we think the particular research were thinking they were related to cellular malaria. And because these kind of samples are quite unique, are very hard to get hands on it, so we had to make the best out of it. So we use the clamp so you can observe the same sample in both light and electron microscopy. I'm a microscopist, so I love imaging, taking pretty images and get them, for example, in the cover of journals is quite amazing. But an image is more than just a nice or a cute photo. So we can use different softwares to do image analysis. So you can improve the analysis out of those images that you are taking in the microscopes. So we have different workstations that have different software packages that are available for the users, and we give them support in both using measuring the, for example, fluorescent intensity or tracking cells that are migrated through a particular piece of tissue, build 3D models, or even build pipelines that can do automatic image analysis for them. And with that, I would like to acknowledge and say thanks to all the users that allowed us to show some of the data here in this presentation, as well the funding bodies that have directly or indirectly support the acquisition of these systems and the maintaining of them. And with that, I finished with my contact and the general inquiries for the MVLS or for the SRF. And please feel free to email me whenever you want to discuss how we can support your project.